All right. Can you believe we're in the last hour of this? And I had the privilege of hosting the first panel, not the first hour, but the first panel. And now I get to host the last one. And there have been some great sessions this time through. And it's really been, just an absolute privilege to be part of this. But here we are. This is the last, and as Katy said, last but not least, and that's maybe an overused phrase, but I think in this case it really applies. This is a great panel, and we're going to talk about visions for the future. And so what I'm going to do is I'm going to just introduce the four panelists by name, and then I will give them a fuller introduction, and they can do a short presentation. And then at the end, we'll get together and open it up to a general discussion, get questions from the audience or questions from each other that arise. But so welcome to Gary Bader, Zorina Gallis, Michael Snyder, and Katy Börner. And welcome to the panel. So Gary, I see you're there, so I'm going to put you up real quick and introduce you. Let me share my screen with you.
And I see Michael Snyder on here also. He might have to turn on his camera to be visible to everybody. Good morning.
Morning, sorry about that.
All right, so Gary Bader from the University of Toronto, Canada, professor at the Donnelly Center at the University of Toronto. His research topics include computational biology, systems biology, network biology, cells in development, regeneration and cancer, and single-cell genomics. And he runs the Biological Systems Lab at the University of Toronto. He holds a Bachelor of Science and PhD in Biochemistry and Computer Science. And he is on the Human Cell Atlas Organizing Committee. I think we've had some people from the Human Cell Atlas, so it's good. It's nice to have you here. And Gary, I can either just simply advance the slides as you tell me, or you can present from your own. Let me know what you'd prefer.
I think it's probably easier for you to advance since you have all the slides. So I'm just going to give a brief introduction of what we're thinking about in our research program. So the traditional focus of our research, the research in my lab, has been mostly thinking about how the cell works and how it fails in disease. We can go to the next slide. And so this is, and we've approached this using a systems biology sort of paradigm or way of thinking, and the general ideas that we want to think about biological systems and how to use those biological systems to understand biological functions or for instance aspects like how the phenotype is how the genotype predicts the phenotype. But we would do that by, instead of just looking for correlations between genotype and phenotype, we would try to take a mechanistic view and understand how systems are affected by genome changes and how those systems then explain phenotype changes. And where we've been involved traditionally also in developing a lot of community resources like Cytoscape for network and visualization, and Gene Mania for gene function prediction, and Pathway Commons for collecting all the information about pathways in the cell. And this is collaborative efforts with a lot of other people: Triadiker, Chris Sander, Quade Morris, Emek Demir, and the National Resource for Network Biology, which is an NIH-funded initiative for promoting this field. Next slide. However, so that, I've focused my lab research on that for quite a while, but what we're thinking about now is kind of moving up to higher scales of biology, enabled by being able to measure more information about these higher scales from single-cell genomics measurement technologies. So the new research focus that we're thinking about is more not just how the cell works and how it fails in disease, but how multicellular tissues work and how they fail in disease. Next slide. And so, I think that's an interesting future trend for biology. A lot of biology, for instance, cell biology is, even the name cell biology is really focused on thinking about all of biology as like a cell, the cell. We have textbooks like Molecular Biology of the Cell, We call things cell lines. There's an idea of a cancer cell, the cell atlas. Everything's very cell oriented, and there's this idea that it's just one cell, and we're trying to understand how the cell works. But a new systems biology could be, let's think about ecosystems of cells, and it's instead of systems within cells, it's systems of cells. And then a lot of additional questions arise, like how do cells work together? What patterns are implemented? What are the functions of these patterns? For instance, if we just think about the human body, one of the important functions is wound response and repair. So if you get a cut on your skin, lots of cells come together to fix that cut. And that's a function that we can think about. And we can, describe that function like we would describe gene function. People spend a lot of time thinking about gene function and functions of systems in the cell. And now I think we should spend, a lot of time thinking about functions of multicellular systems and how they work. So, so that's the kind of thing that we're thinking about now. That's it. Thank you.
All right. Thank you very much. And let's go now to the next slide. And
can I drive the slides, Todd?
yes, you can.
Right.
Let me just I'm not seeing your bio in here.
Okay, can you see my slides?
I can yes, I can see your slides. Oh, there we go. Okay. So, thank
All
you. Let me just introduce you. This is Zorina Gallis. She's interested in cardiology, biomedical engineering, and vascular medicine, and in the realm of pharma, cardiovascular drug discovery and development, and part of the Government National Institutes of Health and the National Heart, Lung, and Blood Institute. And I like that. Make science happen for people through people.
So thank you very much. So this is actually not just my interest. I worked in all those specialties in academia, pharma, and I'm currently working at the Heart, Lung, and Blood Institute, which is one of the National Institutes of Health institutes. And that's my professional motto. And I think I like to put it up there because a lot of people are asking me, are you jumping around a lot? You've been in all these areas. And actually, I was originally trained as a physicist. So I then pursued training in cell biology, biophysics, vascular medicine, pathology, and so forth. So I think that we need to bring all these specialties together in order to be able to answer important questions. And I chose to make myself, let's say, a guinea pig for going through this continuum of specialties. But in order to realize the big vision of the future, we actually need a lot of experts and a lot of artists, as it will become apparent. So the reason I got into mapping to begin with is because I have been pondering on enduring medical mysteries for the past decades of my career. And again, being trained as a physicist, it occurred to me that it's important to find out if there are common mechanisms or sort of guiding principles that may work in such different conditions as shown here, and just some of them are shown here. Conditions that hit us from head to toe and from cradle to grave. Many of them have as common denominator vascular or even more specifically endothelial dysfunction. And so it seems that potentially in order to come up with a solution, we could concentrate on what's common among these diseases. The questions that we have to ask and answer are multiple. Obviously, the vasculature spans the entire body. So which blood vessels, what type, where exactly, why, who gets the disease, when, and how. However, the way we deal with complexity is by delving into complexity, and that's not only the biologists, but even the medical professionals. So, for instance, if you suffer from a systemic disease such as hypertension, you might have symptoms in various parts of your body, but if you would like to do something about them, you might have to go and consult with many different specialties, from neurology to nephrology, and obviously cardiology being what's generally considered a vascular disease. So in order to deal with the complexity, we develop these knowledge silos. That means that many of us spend our lifetime training, specializing, attending different type of professional meetings, and even applying to different institutes for funding. So many times these specialists don't get to talk to each other, even when they are all interested in actually the same system, the vascular system. So we put together a number of professionals from the National Institute of Health, from many different institutes, and you don't have to understand the acronyms here. Suffice to say, for those of you who don't know, the National Institute of Health are parsing out budgets and interest and research based on organ systems or diseases. So again, from the brain research to eye research to kidney research to heart research, we are all working in different institutes, but we all came together and made a proposal to the NIH directors. The NIH has this wonderful program that's called a Common Fund for questions that cannot be answered by one institute or one specialty only. And in 2015, we proposed that we would like to develop a map of the entire vasculature where we can really tell how this functions across the human body, across a lifespan, from development to aging, as well as in various diseases. So in trying to explain this, which is a pretty complex concept, that we want to go from microscale, and here's a single cell, an endothelial cell, to tissue pattern of vasculature, and all the way to the big issues that develop in the large vasculature, as well with incorporating genomic information. It's almost like trying to get a Google map of let's say I want to go to the White House. I can look up the address, I can look up the neighborhood, but I could use my phone, and the phone will even ask me, what is your perspective? Where are you starting from, and how would you like to get there? So in an intelligent way, it can put all this information together and actually give you some very good pointers. So we said we would like to have a Google map of the human vasculature. This created a little bit of a pause, but then we immediately got the request, can you actually build a Google map of the entire body? And this is actually how I started my let's say adventure in mapping. And again, it all started with the desire to be able to ask very important biological and medical questions. This led to this wonderful program you heard a lot about, which is the Human Biomolecular Atlas program. And we are at the step where we're developing technologies. We have established tissue mapping centers that look at various organs and tissues. We are really very interested in collaborating with other consortia across the world and across the human body in order to be able to put together all this information and say something about how normal human cells at this point are collaborating, and you just heard from Dr. Bader, to produce the function that is essential for the human body. But since we are looking at visions of the future, I think a very important thing will be to develop towards the functionality of these assemblies of cells and tissues and organs, and be able to answer very important medical questions. So we are continuing to pursue the idea that the vasculum, which is a concept that integrates not only the physical representation of all the vasculature, but all the information that comes from the many new technologies, the many omics, in order to answer such questions. Specifically for the topic of today, the vasculum is useful for mapping. We propose that it can provide a body-wide cellular positioning system that will allow to map all other cells. And we just heard from Dr. Weber and his team their wonderful progress in putting together the vasculature as a common coordinate frame for the human body, because we believe that this is a nature-inspired solution. Basically, nature knows how to reach every single cell in the human body to bring the nutrients that are necessary, including oxygen, and also is a highway for communication. So all the secreted factors travel through the body from various cells and tissues and organs. And so this system can also be used to target various cells, as well as to report on the function of various tissues. And I'm going to say a few words about that. So I was asked to contribute with some thoughts about the future. So one futuristic thing, let's say, that's happening right now, and I would like you to see the date here. In 1935, it was proposed that the eye is such a personalized window, not only in the soul of criminals, but in their body identities, and cannot be changed, such as fingerprints or faces. And so they could not hide if we found a way to really map the vasculature of their eyes. And this is a recent depiction showing various blood vessels in the eye of an individual and the way you can actually analyze them. So this is a very individual feature that nowadays actually has been developed into being able to say a lot about the person that is being examined just through this non-invasively, just through this window into their body. So you can not only say something about their size, body size, gender, but you can also tell things about their health, whether they are smokers, whether they have high blood pressure, they have diabetes or other conditions. And this is based on the fact that this tissue is fairly complex and it has a lot of different cell types. And obviously, I'm going to point out one of the important cell types here is the endothelial cell, which is the cell that lines all the blood vessels. So basically, looking into the eye of a person, you can look at the status of their vasculature throughout the body, as well as the status of their brain. So this is already something that is happening now, and it's revolutionizing diagnostic and potentially medicine. Another window that has been open for medical applications is the skin. So here's an image showing how you could image the vasculature under the skin of a person and figure out whether they are good candidates for operation based on their the health status of the vasculature. This is a very recent report. I'm thinking maybe last week only, where people have from University of Texas, investigators from the University of Texas, showed that you could measure blood pressure by applying these tattoos that are removable, but they can be used to continuously monitor the inside, basically, status of this person. And this is a publication that is coming from HubMap that is showing how exposure to sun modifies the location and the composition of single cells and specifically immune cells here. So it comes from the G group that is led by Dr. Ginty. So this may remind you of something. If some of you are Star Trekkies, as you may remember, everybody got diagnosed just by using this tricorder. And funny enough, you can buy one on Amazon. I don't think it can do this yet, but it's a wonderful reminder that people already have dreamt of such ways of diagnosing disease. Now, the next window, I think that's just about to be open is the vasculature. Remember, this is how we got started. And the reason I'm so big on this, it's because the vasculature can report on the function of the all the organs, and specifically the functional tissue units. And you already heard people referring to them. And my specific, again, trying to reduce everything, the complexity to one thing, is that the endothelial cells in their entire coverage of the vasculature are able to have local specialized structure and function, such as shown here. So these are blood vessels through which we perfused a red dye. We developed a mouse that was showing fluorescence in all the blood vessels, and the dye is staying within these vessels because they are lined by very tight endothelial cells. These are essential for the functional tissue units of many organs, including potentially here shown the lung. The important thing is that these cells can be recognized by their zip codes, and these are found through a single cell analysis. So each of these cells are displaying their addresses, so things can reach them and enter the tissue based on such markers. In the liver or the kidney, you have different types of endothelial cells, discontinuous, and you can see the red dye is now leaking here because in the liver and the kidney, you need to have very high levels of filtration of things from the blood, between tissue and the blood. And again, the functional tissue units have as a key component endothelial cells. So being able to characterize endothelial cells at the single cell level and their interactions with the cells around them, I think will create a barometer of every organ function. If you can imagine, each organ is a collection of many of these units in order to provide enough function as is needed for the size of the human body. You realize that by getting that signal, we will be able to say something about the function of the organ. And importantly, I think for many of us, we may be able to get very subtle changes in the function before you actually have full disease. So, of course, people ask, so how are you going to get into this window? This window, with the exception of what's really visible from the skin, is pretty deep. And of course, help came from a potential help came from the discovery of the nanomachines that I thought, well, you could send the nanomachines through blood This is a depiction, an artist's depiction of how you could send such a little machine through the vessels and discover function. And of course, somebody said, but you know, this was actually something that was envisioned by a movie in 1966. And as you can see, the difference between these two dates is very big. So the artists are actually helping us dream the future. The artists have not only the capacity to dream, but they have the capacity to convey their ideas in a way that anybody can understand. And then as scientists, we need to be able to catch up and be able to actually bring these dreams to the future. So I want to thank Dr. Börner for coming up with this great idea of even calling this the art and science of mapping. Thank you very much. I'm looking forward to your questions.
Thank you so much for those windows into the future. And I think it's great that Fantastic Voyage, which made an appearance in the very first hour of the 24 hours, has now made a full return. So that's great. Our next presenter is Mike Snyder, and I'm just going to share my screen here. All right. So Mike is a professor at Stanford School of Medicine. He's the chair of genetics, director of the Center of Genomics and Personalized Medicine. His research topics include precision medicine, multiomics, big data, single cell maps, and technology development. And of course, as many of you know, who've seen the VH and things like that, he's also a HubMap participant. Somewhat famous for his kielbasa and hot dog demonstrations. So Mike, take it away. I can just advance the slides for you.
Sounds good. All right. Well, thanks as well for having me here. I think I stem nicely from Zorina's talk. We're one of the HubMap participants, as you just heard, and our mission relative to HubMap is to build single cell maps of the intestine, human intestine. And I might just say I'm also a member of the H10 consortia, where we're actually using this to look at early stage colon cancer, building single cell maps of that. And so I put these together quickly, but I want to just say some of the key people at our consortia: John Hickey, as well as Winston Becker, Will Greenleaf, and Gary Nolan. So next slide. As I say, our map is our goal is to map the human intestine. Turns out the human intestine, you may or may not know, it's super complex. It has all kinds of different cell types. You have more immune cells in your intestine than anywhere else in your body. And so this top is a simple layout, but this 22 feet of hot dogs is very complex as you go from left to right with many different sections with very specialized functions. And then you go into the colon, which large intestine or colon, which is about 7 feet and loaded with microbes, and actually is really critical for fermenting and digesting and making essential vitamins from your microbiome. So very, very complex organ. And so our mission is to build single cell maps of this across the intestine. And so we've been using a variety of methods, Codex, 10x for doing single cell mapping. Actually, we do bulk proteomics and metabolomics and such as well, and trying to integrate that information. And the next slide, I'll just tell you about our Codex work from John Hickey. So we've done Codex now across 9 donors across these different regions of the colon, and we can map out the different cell types like the picture I just showed earlier. And what we'll do is we'll see what cell types are where. And this just gives an example. It turns out the cell types vary quite a bit as you go across the intestine from the stomach left to all the way to the descending region of the colon. And we'll see these different neighborhood. There are these different cell types, if you will. And it turns out they're organized somewhat differently as you go across. Next slide. We call these neighborhoods, the cells that they'll come together in different groups, and that grouping will vary as you move across the colon. And that probably reflects the specialized functions of these cells as you go across the colon. And you can even take this further. You take these neighborhoods. So these are the different cell types and some of the immune and stromal neighborhoods. The next slide. it shows examples of these neighborhoods. You can actually see where they sit in different regions of the colon. And believe it or not, you can take this one step further. You can actually get neighborhoods of neighborhoods. We're calling those communities. So we're really trying to build, if you will, the single cell organizational maps and see how they differ spatially and then functionally across the intestine. That's the flavor. And along the way, as we do 10x and these other things, we'll discover some new cell types where we'll see, for example, we found something called mucin 6. That's an interesting a molecule, the mucin 6 producing cells that's in one region of the colon and so on. And then, as I say, we're also using this as a template as part of H10 to start understanding what this organization looks like in early stage cancer. So that's a flavor of what we do. We are funded by HubMap. We're trying to build 3D maps of this. I want to save time for the discussion, so I was just going to introduce this as this, and I think I'll just insert my vision as we get into the discussion part. So that's kind of what we do. Oh, I should say we do a lot of gene regulations and try to understand, how the critical regulatory steps involved in producing and organizing these different cells as well.
Thank you. And next up is Katy, and I think she's going to share her slides, but I did want to just share this one with you. This is her intro.
Can you see my slides?
Oh, yeah, there you go. You've got it up. Okay. Katy is the Victor H. Ingve Distinguished Professor of Engineering and Information Science at the Luddy School of Informatics, Computing, and Engineering at Indiana University, where I also work. Director of the Cyber Infrastructure for Network Science Center. Curator of the International Places and Spaces Mapping Science Exhibit. Check it out at scimaps.org. PI of the IU mapping component for the NIH HubMap and SendNet. Member of the Atlas teams in KPMP and GoodMap 4. And she holds an MS in engineering and PhD in computer science. And this is a handsome collection of people. You want to say something about those?
It's a team effort. Yeah, I'm just presenting the work of many, many smart and diligent and passionate people here. It was wonderful to see actually the life trajectories and to hear more about how people got attracted to mapping the human body. And maybe that requires an entire other 24 hours event. But in the interest of time, let me just tell you a little bit more about how many people it actually takes. So here on the left-hand side, you see different consortia around the globe interested in mapping the human body at single cell resolution. And those are not even all of them. SendNet was just funded. H10 that Mike Snyder is part of is listed here. But there are many, many others that are really now just getting started on this. And here you see a list of organs. And if you only have those organs, you're not going to be surviving this. So there are many other organs that we might also like to bring in. And there are now first papers with many, many authors, not yet thousands or 10,000s of authors, but I could very well imagine that the final first draft of a human reference atlas has 1,000, if not 10,000 authors. And what we are trying to do is to create a community and infrastructure and user interfaces so that experts from very different disciplines and very different scientific cultures and environments can collaborate on this together. This is a socio-technical engineering problems that we are trying to solve. And there are many data portals, including the HubMap portal that you see here, which you can now explore. And you saw beautiful other portals in the previous panel by EBI, by Cell by Gene. So all of these hold a lot of the rich new data that is beautiful and ideally can be cross-queried soon. In HubMap, we have 26 assay types and more than 1,000 tissue samples. These are biopsy data sets or larger tissue blocks that get cut from human tissue and then get analyzed. And it's non-trivial to interlink those data sets across scale. Here you see the entire human being at a meter level. Then there are, let's say, 10,000 anatomical structures that we care about. And then we know that there are about 37 trillion cells. So it's a huge number of elements that need to be spatially characterized also named, maybe with cryptic names, potentially machine learning generated names, because I don't think we have currently these 10,000 anatomical structure names. And ultimately, that data need to be linked. This here on the left-hand side is the experimental data. On the right-hand side, you see the evolving coordinate system, initially just in 3D using the Visible Human Project data that many other teams have used over the last 20 years. But going forward, also using a vasculature-based reference system that Griffin Weber and his team just presented this morning. These microenvironments that Gary and Zorina and Mike just mentioned are really important because they help us get from the organ level, which is like 10 centimeter or rather sizable, and coming down to a much smaller scale where you have 1 million glomeruli in each kidney, and down to the single cell expression, here gene expression levels. So here you would have Asimov's references or cell typist references as we had presented yesterday. And the way to potentially federate all this data together spatially is to, first of all, register it into one coordinate system so that we can spatially query that data. And so that is now possible. And actually, as you move that yellow tissue block into a reference organ, you get automatically different anatomical structure tags assigned. So you can use them to query for that. And then you can see what tissue blocks now exist in which areas. You can then zoom into these functional tissue units, and we now have 19 across 9 organs where we have schemata in 2D, not yet 3D, but 2D is great for having experts agree on names and shapes and sizes and general number of cells per cell type. As 3D data becomes available, these will become 3D, but right now that's not a data type and set we have yet in the number that we would like to see. And then you will see gene biomarkers, protein biomarkers, lipid biomarkers for specific functional tissue units. And you will have different source type data, maybe from young people, from mid-aged people, from older donors. And you will be able to click through those and you will see what changes in terms of biomarker settings across age or across BMI or sex or ethnicity. I think it's really, really important to capture that human diversity. So we have to keep track of specimen data also, not just spatial and the biological structure, but also the specimen data. And then, of course, you can explore the provenance of all that atlasing data. And here you see the Vitesse viewer that is integrated into the HubMap portal. And you can click on different tissue sections and just explore that in more detail. Going forward, you can then find your own data in these data sets and see what other data sets are close by. And Zorina also already showed that we are looking in understanding the distances of different types of marker genes for disease or for here immune cells to not just the surface of the skin, but also to vasculature. And ultimately, we need this kind of data in 3D for multiple organs. Right now, we have it only for skin, thanks to the collaboration with the GE team and the University of Pittsburgh RTI. And then you can go from the 3D spatial registrations of tissue blocks to these ASCT plus B tables, to the vasculature distance distributions. And again, we need this for multiple donor populations, demographics, and for different organs so that we can truly understand human diversity and also what happens across organs and what's very specific to each organ. Now, you might wonder in how far machine learning can help. And I used DALL-E recently to get us all a seamless textile pattern design of geometric human anatomy atlas map in digital art. So if you add this into DALL-E, you will see this rendering on the right-hand side where DALL-E frantically tries to interconnect all of our organs using the vasculature. And of course, it doesn't have much good training data to go by. And I think this is also the wrong level that we should use for using machine learning. But it's actually quite beautiful. And as you see, it's well labeled. There is no scale bar, however. We have been using machine learning in cargo competitions. And here, I think it's beautiful to engage 1,000+ teams to work together on developing code to segment ultimately also to animate, not just annotate, not just single cells, but also functional tissue units and anatomical structures. And there are 2 more competitions planned in support of VCCF. And in the interest of time, I will leave you with a paper that explains how we construct the Human Reference Atlas within HubMap, and how we can then use the Human Reference Atlas to map new data into it, but also to run new queries, queries for where is the most highly expressed gene, where is my favorite gene, where is the favorite cell types that I'm working on. So feel free to check that one out. And for those of you which are totally new to single cell studies, check out the Open Data Code and Training in the Visible Human MOOC. And I will give it back to the panel moderator. But keep in mind, the Human Reference Atlas is on a 6-month release cycle. And if you want to learn more, just come to humanatlas.io and you will learn a lot more about this atlas effort. Thank you all.
Thank you, Katy. To the attendees, those out there, please put your questions into the chat. If you have questions for the panel as a whole or for individual members of the panel. The first one we have here is one for Mike from Katy. Do you think that human-identified FTUs, functional tissue units like the 19 in HubMap, will be rediscovered through machine learning segmentation algorithms? How many others might those algorithms discover? 100, 1,000, 10,000?
Good question. So the answer is, of course. For one thing, we actually do our training on known cell types. So we have methods now. There's an algorithm called Stellar that John Hickey, together with Yuri Lesovic's group, built that actually does that. Not only takes known cell types, annotation from known cell types, and can put them on new data maps that you're generating, but it actually can define new classes of cells, which is pretty cool. We don't necessarily know what they are, but you can see here's a new subcategory. I think this all comes down to how big of a splitter you are, how many new cell types you'll find. You can always find more and more classes as you dig in, and it'll get to be very interesting, the definition of what's a cell type versus a cell state. And so I think that'll be an interesting philosophical argument. But yeah, so the point is we will see more. And I think this relates a lot to Gary's early comment, which is we will want to put together not only just cell types, but also cell states. Cell states very, very important, both from an environmental perspective, but from a neighborhood perspective. Who you're interacting with determines your behavior. You see it in humans all the time. People act very differently in different contexts, and certainly in the influence of what they consume. So I think defining all that, it can be very, very useful for understanding how organs and cells work.
Thank you. So as the audience formulates some questions, I would just open it up to anyone have questions for any in the panel have questions for other members of the panel, or any ideas that were sparked by folks' presentations. Feel free to share those now. It looks like
I'll throw this out to the other panelists, which is, I assume what we're going to talk about is vision. Where do you guys think we're going to be in 2030? What's this going to look like? So,
great question.
I wanted to just, maybe address that point. And first of all, I forgot to mention earlier, it's great to see this 24-hour initiative, and thank you for inviting us and organizing it. And really a lot of respect to everyone who stayed up for the whole 24 hours. I watch periodically, and I also love the interdisciplinary nature of the talk. So it'll be cool to see the videos. But I talked a bit about trying to better understand the function of cellular ecosystems. And I think Kathy's calling them functional tissue units, or FTUs. So I just wanted to kind of talk about a vision for where that goes a little bit, just one aspect of it. So biology is inherently multi-scale. We have SNPs, and we want to understand their effects on human populations. And obviously that goes through a lot of scales. And so I just want to explore this multi-scale idea a bit. So people have been thinking about multi-scale for a long time. When I was searching yesterday, I noticed this I remember this Powers of 10 film, which you probably have seen from 1977. I didn't realize the history of it. Design, it was made by famous designers Charles and Ray Eames, and I didn't realize it was made for IBM. So it's interesting. It's a nice historical example of an influential integrated thinking between science, technology, and art. And if you haven't seen it in a while, you should check it out. I'm just going to paste the YouTube link. So interestingly, this film covered from the universe to systems and molecules inside our body. But what it didn't do is it didn't comment on how to think about multi-scale systems. It just showed it to us, which obviously initiates thinking. And there's lots of multi-scale systems, but I'd argue that there might be some important conceptual differences between them. So for example, physics has long considered multi-scale systems, and they've been analyzed in all sorts of ways, or even simulated a high level of detail, like nuclear reactions and galaxy simulations. So we definitely want to learn from physics and other areas of multi-scale studies, but I think that biology is different. So an interesting thing about biological multi-scale systems is that they're evolved. And I'd like to argue and maybe discuss that it's an important conceptual difference, because in evolution, we copy and mutate. And that means that biology has a lot of repeated, copied, and evolving patterns. And what's interesting now, 2022, is that machine learning is so powerful, deep learning for finding automatically the patterns, right? And Katy already just kind of mentioned this. But so I'd like to argue that an important future activity will be to use machine learning to recognize reuse multi-scale patterns in multicellular tissues. And then once we identify these, whether they're cell types or states or gradients of cells or all sorts of patterns that we don't even think about yet, when we look at these UMAPs, then we can interpret them to understand their function. But we'll want to understand them from an evolutionary perspective in terms of the entire pattern and how it's repeated. I think you can think about that in all sorts of different ways. So I wonder if that's an interesting kind of one of the maybe conceptual frameworks that we can use to think about where we're going to go in the future.
I put the comment there, so I agree with everything Gary said. I put a comment that given the fact that the current technologies, so time, resources and everything are not going to let us map every single cell of the human body. Again, as a physicist, I would come to what's the essence of every organ. So what is the function of the kidney? What is the function of the lung? And if we can map and explain functionally the function of every organ, and we can put together these many repetitive units that are helping that function, we can already have the first version of the human body map. That's something that I proposed several years ago, and I think it's beginning to happen with the help of Katy and many Gary, and I think we are usually evolving in waves. So it's the wave. I think it's building, and I think again because of these repetitive patterns, the machine can help you fill in a lot of things based on what should be in there. But again, I'm coming back to my very old saying, and people who know me are very tired of hearing, but pathologists already have this capacity of looking at any type of tissue section and recognizing the pattern. So if the human trained eye can do this, I'm pretty sure the machine can do it even better. But the most important thing is, what are we going to get out of it? And for me, that's the function or / dysfunction of what we see in this map. And it doesn't come from just one cell, but from groups of cells doing things together. So, and that's the first, and I hope it doesn't take until 2030, Mike. I hope that we will have.
Well, no, that I think can be
soon.
done much earlier, especially at the resolution you're describing. I think I was trying to push, especially when I have 10 minutes left. Where do you want? Where should all this go?
to Star Trek, I envision that we are going to know everything about what's happening in the body from the signals that we are able to interpret from deep inside the tissue.
Mm-hmm. Yeah, I would argue that actually, kinetic maps with which we don't have technologies for, not just making a static map, but a dynamic map where you can actually see things. Certainly, people are working on developmental maps by looking at different stages of, fetal tissue, that sort of thing. But one could argue to really get a functioning map, you'd like to see the kinetics, and there aren't technologies for that sort of stuff, especially in humans, obviously. You probably can't do those sorts of things, maybe on the skin. But yeah, certainly for animal models, if you really want to get a function, adding quantitative dynamics will be pretty powerful. So that would be an example of something that I could see possible. We'd have to vet some new technologies to be able to do it. So love to hear from that's just one of many ideas, I'm sure, that are out there.
One idea that came up in the collaboration with the GE team was to try to have a digital twin, a parameterizable model of the human body that could be coming into existence as you are born and then grows with you. And every single time you have your checkup at the doctor's office, there are new urine and blood values that could be used to parameterize it. I know, Michael, you have all of your tracking devices on you. So all that data could feed into the digital twin that is your mini-me, so to say. And then you have a healthy reference and you know what you had before some disease got you. And you could compare across, and that would be very, very valuable. But then, of course, we need 8 billion of these mini-me's. And do we even have the compute power on this planet to actually do that? And maybe that leads over to some responses that Gary recently had when we got asked a similar question. And maybe, Gary, you want to take that one?
Yeah, I guess, Katy and I were at a workshop and discussing some of these ideas earlier this year. And, there's if you do want to have a digital twin, how are you going to implement it? And someone asked us this question, well, it's hard to do that if you're going to simulate the galaxy. There's so much computational needs, for instance. But I think that these patterns are actually going to help us because we're going to find out that we can reduce the dimension of all of our measurements to a much smaller set of factors, and those factors are going to help us understand the kind of rheostats of the body that are going to be the key variables that are being played with over evolution and during physiology and the kinetic, the dynamic, temporal aspects that Mike mentioned. And once we understand some of those fundamental rheostats, we'll be able to implement those much more simply computationally in a digital twin type of thing. So I think that's one of the hopes, is that we'll be able to discover these fundamental factors. I don't know
maybe that leads us back to a discussion we had in the 9 p.m. panel on human body exhibits, which had a lot of artists in there. And they were trying to figure out what attracts attention, keeps attention, and makes people act differently, right? So if you're in a science museum, you can't teach, you can only engage. And there's a lot of stuff going on in each science museum. So why should you now be interested to learn about how to keep yourself healthy and happy and sane in this very complex and pandemic world? And it was interesting to hear the comments on how to best engage and how to change behavior so that we all live healthier, because in many cases, we know what to do to be more healthy, to be more more sport, more greeneries. It's all known, or many of these things are known. And how do you make people act on it? And then, of course, Mike would have many more comments on how to behave better, because he, again, is doing this extensive body value tracking. But to bring this to large numbers of people so that they all live healthier and happier lives is non-trivial. And I think we saw this with the rollout of the vaccines and with other elements.
Well, maybe I'll jump in here. I think there is a big push these days, partly because of financial reasons. People are, a lot of billionaires are investing a lot of money for longevity in that general space, longevity and health span space. And so there is a lot of activity. I think that does create awareness. How much of it gets to certain regions of the country? Well, that's going to need a lot of work, but there's no question there's a segment of it that actually adopts this pretty quickly. And then it may take time, but I think it will diffuse to the entire population. Also, as the population gets older, right? Everybody born today is going to live to be 100, or it certainly has that capability. And so that's going to shift the dynamics of people's thinking. And I do think that as a consequence, people will get more concerned about their health, at least maybe this is wishful thinking, but that should lead to this. And I think these sorts of reference maps, of which this is one, paradigm of that, would be very, very valuable for that. Yeah. By the way, all the healthcare companies are talking about digital twins. Some of them are really doing it. Some of it's just, buzzwords. But
I was going to say that closer to us, we could already have cellular twins, right? So you can extract cells from individuals and put them under certain conditions and see how they would respond to different things, and whether there are certain mutations or defects they already have that are going to be expressed at some point. So just that's kind of a more tangible, more immediate future. The iPS cells. Yeah.
I have one more thing to mention. I was just reading a book about quantum computing, trying to understand it. I think it was called Schrodinger's Killer App. There was a mention there that there's a lot of military interest in this and that technology. And so one of the things that the military does to create visions of the future is they have reportedly an annual meeting where they invite mostly science fiction authors, and then they just listen to all the interesting ideas that pop out of getting a whole bunch of like 200 science fiction authors together, and then they try to record interesting things that might actually occur to think about how they would deal with it. I wonder if we should do something similar for biology.
There are entire institutes which do that, right? There's the Institute of the Future, and that's their job. They invite relevant people. And yes, in some cases, you invite industry experts, and others you invite science fiction authors. And then you create a report and very systematically so, right? And in Japan, this is done so systematically. You can actually go back in time and see if the predictions they made 10 years ago actually came through, and which ones did fail and which ones did come through. So it's a very systematic forecasting process, actually. Happy to connect you to experts there, and I would love to be a fly on the wall and listen in.
I mean, Zorina kind of mentioned it in her presentation, also referencing science fiction quite a bit.
Mm-hmm.
So I think, as I said, not only they dream the future, but they are able to say it in a way that engages everybody at all levels. And I'm not sure whether we are usually just trying to catch up with them. It's like science imitating art, life imitating art, rather than art imitating life.
Thank you.
Yeah.
Oh, I'm sorry. Go ahead.
Science informing art, art informing science.
Okay.
True Renaissance engineering, yes.
Thank you so much. This is a great note to end on. And yes, thank you, Zorina, Gary, Mike, Katy, and wonderful panel. And I think that Katy take it over, and we've got an official send off and maybe some thank yous.
Yeah, thank you all. Thank you to the panelists, which were brave enough to come into a panel on the visions for the future. And of course, we want desirable futures, right? There are many dystopian futures which we might not like to have. So it's always good to kind of project a few forward and then pick one that's actually desirable. In addition to the panelists and also to Todd, who was heroically moderating many of these panels. Thank you, Todd. We would love to thank also Todd again for compiling the video collections that we're running in some of the late night hours. We would love to thank Melanie Goldstone, Bruce Hare, and Bushan Cope for designing the website, Devin Wright for media support and help keeping organization of the organization of the event. We would love to thank Martin Matthew and Tracy Theriault for advertising this event, Andreas Bückle and Devin Wright for making this recorded Zoom event smooth and safe. Wasn't totally easy, but I think we managed that well. And all the video recordings will be made available on the website. So check back in a week or so. I think it's really cool to actually look what other panels had to say. There's a lot of synergies across. And now I can also onboard students more effectively because I can just point to the details on Cell Typist, for instance. Andreas Bückle was here for the professional media setup and great panel at 10 p.m. last night. Monica Herzig for providing great jazz music for the beginning of this event. And then also thanks go to Melina Casadio at Nature and Alex Bannister at AppCam for social media support. And funding for this entire event was kindly provided by Maury Goldstone, the late dad of my husband, and CNS and Science at IU. Thank you all.
Thank you. Goodbye.
Thank you. Bye.
Bye, everybody.
Thanks.